Guarnizo, and I'm going to talk about today about geographical factors promoting diversification of the Northern Andes and Brazilian Cerrado regions. The case of frogs and Anole lizards. So we all know that South America is a really diverse place. However, not everybody knows that this diversity is not randomly distributed. We have regions with more diversity. For example, this picture shows the alpha diversity patterns of amphibians, and we clearly see that it's clustered around the Amazon region. However, there's an interesting pattern when we see beta diversity of species with smaller geographical ranges. For example, this is a diversity patterns of amphibians as well, but with a short or small geographical ranges. So we see a clear difference, and there are many studies that try to understand this. Clearly, we know a lot of research. For instance, 2 years ago, there was a really influential paper about it called The Drivers of Tropical Speciation that suggested that in tropical birds, for instance, there were two main predictors for the speciation of the tropics. One was the amount of time a lineage has persisted in the landscape. We know that South America has a very dynamic topographic history. We see that the Andes have lifted recently, and we have some other important highland regions like the Guiana Shield and the Cerrado, the Brazilian Shield, excuse me. So what they suggest is that dispersal events have an important role in diversification, colonizing regions, and then with time, those older lineages are going to accumulate more species. The second predictor is the ability of lineages to move in the landscape. So we know there are different traits or characteristics of species that might prevent them to move across the landscape. For example, the physiology, or even parasites, or we've seen many different talks about this, the elevation range of species. So once a species colonizes a place, then depending on how well it moves across the landscape, it might differentiate more or less. So what I'm going to talk about here is I had the opportunity of studying these very interesting regions that has some similarities and some differences, which are the Northern Andes around Colombia and the Brazilian Cerrado. The similarities, both are mountains. They are really, really diverse. They are considered hotspots of diversity. However, they are also very different. The Andes have a more complex topographic complexity. They have higher elevations, and the Cerrado is mostly a savanna. It is very stational in terms of precipitation across the year. So I had the opportunity to study some frogs and lizards in these two regions. So I'm going to try to compare what is going on in these groups. So in the Andes of Colombia, Colombia has three branches of the Andes, and we had the opportunity to do a transect across the eastern Andes of Colombia and study populations at both sides of the eastern Andes. In yellow, we have the western populations, and in the red, the eastern populations, and multiple species that have of the lowlands that are both across the Andes. We find the same pattern. We have western and eastern populations, highly genetically divergent, and this happens for many species of the lowlands. So this suggests that the diversity of this area is highly underestimated, and we don't know much about it, about the phylogeographical or genetic processes of these species of Colombia. So probably we'll find many cryptic species in the future. Also, we had the opportunity to study what happened inside species. This is the rogatoid frog of the Colombian Andes, and it has a very interesting distribution. It's in the eastern and the central cordilleras of Colombia, and we found also that the Andes uplift apparently separated populations around 20 million years ago, which agrees with the beginning of the uplifting of the eastern Andes of Colombia. And also, interestingly, we found that lowlands, like these valleys, like the Magdalena Valley, of this valley formed by this river, and the Chicamocha Canyon here, are apparently stronger geographic barriers than the summits or peaks here. So there's more permeability in the summits and less permeability in the valleys. That's an interesting pattern that has been also found recently in birds of the same area. So what's going on in the Cerrado? I had a chance to study this species called Anolis or Menarops meridionalis, which is endemic of the Cerrado region, and we found first that apparently this species is actually formed by 5 cryptic species, like Fabricius recently gave that talk. That also we have other taxonomic groups that are actually cryptic complexes in the same area. So we found that the divergence time in this group agrees with the geophysical activity of the central plateau of Brazil. So apparently, topography also has an important role separating populations in this apparently less geographically complex region compared to the Andes. And actually, this divergence is way higher, 3 times higher than other Anolis that with similar geographic distributions like Anolis carolinensis in the United States. So what we see is a similar pattern that we also was found in birds, that we have first a dispersal event. Probably this group of Anolis came from Central America, and then we have a diversification caused by the topographic heterogeneity of the Cerrado region, and then later we have dispersal events in the different parts of the Cerrado. Now, we know that first dispersal has a really important role in the diversification of these groups in the Andes, but then what is promoting the posterior diversification? We know that mountains they have a really strong environmental gradients like oxygen transfer pressure, CO2 temperature, UV radiation. So for a frog or a lizard, it's not the same to move across the horizontal landscape than to move across a vertical landscape. And so we had the chance to study multiple frogs in the Andes of Colombia and test what is the role of topographic complexity in the genetic variation of these frogs. So we have, for example, here we found that the role of topography measured as a least cost path distance using environmental variables, as well as the absolute difference in elevation between frogs are really strong predictors of the genetic variation of these frogs. Therefore, topography again shows an important role differentiating the populations of mountains. Here we see also this plot shows is a heat map that shows in warmer colors the higher local genetic differentiation between populations. And here on the right is the Andes, the picture of the Andes. And you see here that in all these species, we see a similar pattern. The warmer colors are in the north, and then we have less genetic differentiation in the south. And what happens is in all these cases, in the north, we have more complex topography than in the south. In the south, we have the Llanos Orientales, which are savannas and flatlands, while in the north, we have the eastern Andes in Santander, the area of Santander in Colorado, which is highly complex in terms of its topography. And we found a similar pattern in the Cerrado lizards. We found that the regions with more complex topography have also, inside those lineages, I described recently, we also find a high genetic structure or local genetic differentiation associated with topography. How prevalent is the role of topography increasing genetic variation in South America? In this study, we compare multiple species sequenced by multiple authors and researchers with multiple genes, and to see what's the role of what's the relationship between topographic complexity and genetic distances controlling for the role of geographic distance. And we found in each case that there's a positive relationship between topographic complexity and genetic distances, so which suggests also that topography in fact has a strong effect. So now we know that this topographic complexity probably is affecting the gene flow patterns, and maybe the elevation and range of these species has to do something with that. Maybe they cannot cross the valleys or the summit for some reason. There might be multiple reasons for this happening. So another question is how stable through time are these, for example, temperature niches of the species? In other words, is there evidence for niche conservatism? We use a coarse-grained approach to see this, comparing the niche models of the species at different sister species that they have in the central and eastern Andes, and we find that with these environments are more similar than expected by chance, which suggests that in fact their niches are conserved to time, at least the thermal niche at coarse grain, of course. And we found the same pattern in North Meridionalis in the Cerrado region. So at least in these few cases, we might suggest that there's evidence for niche conservatism, and this, in other words, these niches are conserved to time, and since they are narrow because we don't have any seasons across the year, then that might be related with the Janssen hypothesis that suggests this. So to conclude, we have that the genetic divergence per unit of geographic distance apparently is higher in mountainous regions in South America. It's very interesting because the Andes and this area of the Cerrado and also the Guiana Shield are really highly diverse. Such areas seem to contain a high proportion of cryptic species that have not been described yet. In other words, the diversity of these regions is underestimated. And some of the species we found that valleys and not summits seem to be stronger barriers to gene flow. And finally, it seems that the ability to move across environmentally heterogeneous regions in the tropics is restricted by the narrow and conserved niches of EU niches. So with that, I would like to thank these people and funding. Thank you very much. Any questions?
Thank you, Alice. I'm surprised that valleys and not summits are the barriers. Have you got any sort of insights or ideas about why that might be?
Yeah, that's an interesting question. I was wondering why this might be. There are some studies that show that evolution to lower temperatures is more difficult to higher temperatures. So that might have a role on that. But yes, I was expecting also that higher elevation would be more stronger barriers to gene flow. But honestly, we need to do more physiological experiments. That's what we are planning to do to study the thermal physiology of these frogs, not only in the adults but also in the tadpoles, to see what is going on in terms of what is preventing these frogs to cross these valleys.